Hello dear students. So from today I'll be posting the videos recordings regarding the genetics topic. So earlier I have posted you regarding the chromosomal structure organization, which is also part of this human genetics. So today I'll just give you a brief idea about the eukaryotic cell cycle, particularly human cell cycle, which you have already known and you have studied in great detail. So cell cycle, it includes interphase, mitotic phase and cytokinesis. Interphase further divided into G1 phase, S phase and G2 phase. So G1 is the first growth phase followed by synthetic phase, then G2 is the second growth phase, then there will be division of the cells. So all of you have an idea about this. So these particular cells, they have different cells have different frequency of division. For example, embryonic cells, they divide by 20 minutes, less than 20 minutes. Skin cells by 12 to 24 hours they are going to be dividing, whereas liver cells, they divide one or twice in a year. Even though they have the capacity of division, they don't divide much, only on requirement they are going to be dividing. And whereas some other mature cells like muscles, nerves and all, they are actually not going to be having a division because they are terminally differentiated. They don't divide and they are permanently into the G0 phase of cell cycle, which is nothing but it's a phase of cell cycle wherein the division is not going to be observed in those cells. So the interphase, which consists of G1, G2 and S phase. See, G1 phase is a growth phase 1, in which the time of it's a time for the cell growth. So cell is going to be growing in terms of its size, its components, all those things. And in S phase, it undergoes synthetic phase where the DNA is going to get replicated, components of the cell is also going to be multiplied. Then G2 phase is a chromosomal condensation phase wherein the chromosome, which is produced as sister chromatids in case of S phase, will get condensed into a chromosomal structures, and which is subjected for separation in the next division phase. So followed by the S phase, the sister chromatids appear to share a centromere, and the centromere is going to be replicated, but the two centromeres are held by cohesin proteins, and the proteins of kinetochore are attached to the centromere. Followed by that, microtubules are going to get attached to the kinetochores and start facilitating the all the arrangements for the division of the cell. So this is the structure of metaphase chromosome, wherein you can see the centromere with the cohesin proteins which are attached to that. Then there will be G2, wherein exactly the condensation happens, the centrioles will attach, then followed by that there will be mitosis. Mitosis has 5 different phases: prophase, prometaphase, metaphase, anaphase, telophase. Some people say just 4 phases: prophase, metaphase, anaphase, telophase also. So in case of prophase, all of you know, chromosome is going to continue to condense and centrioles are going to get divided. They move actually to the opposite poles, and spindle apparatus starts assembling, and nuclear envelope is going to get dissolved, so that the chromosomes will start moving towards the center of the cell, as well as the components of the organelles of the cells are also going to start disappearing. They disappear, and in case of prometaphase, which also people consider as a part of metaphase. So here the chromosomes become attached to spindle apparatus by the kinetochore, and the microtubules are also going to get formed, and they develop from each poles to each of the kinetochore, and microtubules begin to pull the chromosomes towards the center of the cell. So alignment of all the chromosomes is going to happen at the metaphyseal plate, or at the center of the chromosome, which is going to be completed at a metaphase conditions. So this is called metaphyseal plate, wherein all the chromosomes are going to get attached to the kinetochore, means spindle apparatus at their kinetochore, and will get arranged in the center of the cell, as which we call it as a metaphyseal phase, wherein the chromosomes are going to be held at kind of tension, because the spindle fibers from opposite poles will be trying to pull apart the sister chromatids. Now you can see this is how exactly the arrangement are going to be seen at the metaphyseal plate, and the sister chromatids are attached with the at their cantochore with the spindle fibers from opposite spindles or the asters. Then next we come across the anaphase, wherein the movement of these particular sister chromatids will takes place as the quiescent proteins are going to get removed, and there will be separation of the centromeres. The chromosomes, the sister chromatids are going to be pulled towards the opposite poles. In the anaphase A, the kinetochore is going to be pulled apart, and B, the poles are going means the uh, chromosomes will be moved towards them. You can see how exactly the chromosomes will appear. It appear as a V shape when it is strained and observed in the microscope. Then we come across with the telophase, where the spindle apparatus disassemble, and the nuclear envelope is going to be formed around each of set of the chromatids. The chromosomes begin to uncoil, and the nucleus appears in a new daughter cells. Even the Golgi complex within the endoplasmic reticulum will start reappearing. Then followed by that there will be cytokinesis, where the cell is going to get cleaved into two halves. In case of animal, it is going to start with the cleavage furrow. In case of plants, it starts with the cell plate. And in case of fungi and protistas, mitosis occurs within the cell nuclei, so division of nucleus occurs through the cytokinesis. So there are two reversible points here. One is replication of genetic material, means once replicated it cannot be reversed, and the other one is the separation of sister chromatids. Once separated, the sister chromatids cannot get attached. The number of checkpoints which we have already studied in previous semesters, so which are going to be checked for the process which is taking place. They are going to be assessed, and in case if everything is right, it is going to allow to continue. If there is anything wrong, then it is going to be stopped. So there are checkpoints at the metaphase. There is checkpoint at the G1 as well as G2 phase, that is between G1 to S phase, S to G1 S to M phase, G2 to M phase, and in M phase also. So these particular checkpoints are going to be controlled by stop and go signals, which we all know they are going to be CDKs. They are going to be cyclins, which are all actually involved in the regulation, maturational promoter proteins, all these things. So these signals indicates the go for the cell division. So the major checkpoints are G1 S phase checkpoint, which is going to check for the growth of the cell, and there is it's going to check whether the cell DNA can begin the synthesis, and G2 phase to M phase checkpoint, which is going to check for whether the DNA synthesis has been completed correctly or not, and it is going to be, if it is found to be completed, then the cell is going to be made to commit for the process of division, that is mitosis. Then the next is spindle checkpoint, that is M phase checkpoint, which is going to check for the proper alignment of the spindle fibers, whether they are properly attached to the chromosomes, they are properly aligned, and the movement of the can sister chromatids separate properly. All these are going to be checked. So in G1 S checkpoint, so this is a restriction point. There will be go signal, which is given by the cell nutrition, extra signals, growth factors. They're all going to be playing important role. If found to be not means insufficient, then it will go to the G0 phase. non-dividing state. G0 is a non-dividing differentiated state. So in most of the cases of human cells, G0 they are going to be in G0 phase. Liver cells they are going to be in G0, but can be called back when the division is necessary or repair is going to be required. So nerve and muscle cells, they are always going to be in G0 phase. So cell is going to divide, the making activation the cells to undergo the division. It is going to happen with the help of some communication signals. In terms of chemical components that we already know, there will be activators, inhibitors. So there are cyclin dependent kinases, CDKs, promoting factors, their growth factors, and dependent proteins. There will be phosphorylation, there are kinase enzymes which are involved in that CDKs and CDK cycling complexes, and they're all the signals which controls the division of cells, which people have already studied well. So cyclin dependent kinases phosphorylate cellular proteins, activates or inactivates and triggers the division of cells. So G1 as checkpoint, there will be mitotic promoting factors, anaphase promoting factors, they're all going to be involved in the regulation. For example, growth factors, nutritions, they are going to act as a first component to check for the signal. If it is sufficient, then it will give a go ahead signal, which is given through the cyclin CDK cyclins. Then the G1 M2 G1 G2M checkpoint, where replication is going to be completed or not, will be checked. So if it is done, then CDK G2, that is MPF, will give the signal for the division. In case during the mitosis, there will be APCs, that is it's going to check for the chromosome, they are going to form metaphyseal plate or not. So then it undergoes the division of the cell. So thank you. So this is all about mitosis. Similarly, in case of meiosis, you people know there is going to be different phases. I will post about the mitosis in the next video.